used to quantify the factors contributing to assembly. All right, Hannah.
Great. So yeah, I'm going to be
Okay.
talking about the framework. Community ecologists really like to ask things like, how do organisms get to where they are today? Next slide. And also, how do new and different communities arise over time? And if these sound very familiar to some questions that Matt raised yesterday, it's because, in fact, we do a lot of the same things population geneticists think about, but at the community level as opposed to the population level. Next slide, please. And so just before we get started, some definitions to make sure everyone's on the same page. So when I say community, I am thinking of an interacting group of species in a common location. And when I say community assembly, I am thinking about the interacting processes that give rise to observed differences in community composition. So if we have like a community, it has different abundances of organisms, what are those processes? Next slide, please. So people have been thinking about community assembly for hundreds of years, in fact, and over those times, we've kind of observed some patterns in community assembly, and there's all these kind of different patterns people have observed, some very famous, like the species area relationship, latitudinal diversity gradient. You may have heard of some of these. And these are patterns, but it's very hard to think about kind of what the processes underlying these patterns are. And next slide, please. Thank you. And this has been termed by many people, and Kelly brought it up before, the black box of community ecology. And in 2010, this guy, Mark Belland, was kind of thinking about these things and saying, man, it would be really great if we could have some way to kind of characterize all the processes that are underlying these patterns that we observe. And so he talked to some population geneticist colleagues and came up with this kind of very parallel way of thinking about community ecology to what population geneticists were thinking about for population biology. And so his idea is that community assembly is controlled by these four processes: selection, dispersal, speciation, and drift. Next slide, please. And the kind of one of the beauties of this framework, as opposed to some of the other frameworks out there that exist for thinking about community assembly, is that it's kind of this like has this ability to be nested because it links very well with this population biology viewpoint, where instead of speciation we have mutation, and instead of dispersal we have migration, but a lot of the same processes happening on different scales. And I tend to think of this as a great lens through which to view even perhaps genes that might maybe mobile genetic elements or homologs within an organism might also be influenced. These anywhere where you kind of have competition of organisms, you might be expecting to see some of these things. Okay, next slide, please. So in addition to having these kind of four pillars, it's important to point out that we can actually kind of categorize or group these figures by, or these pillars by the kind of ability of our ability to kind of predict what might be happening versus kind of the randomness associated with each one. And we call this kind of stochastic and deterministic, whereby some of these pillars, might be very predictable based on species identities, and others might not be very predictable based on species identity. And that's very important. When we talk about stochastic, we're not talking about like totally random. We're talking about random with respect to the species identities of the community. Next slide, please. So I'm just going to go through these four different pillars and talk about kind of what we might expect. So selection we think of as like entirely deterministic, and when we say selection, we just mean the fitness differences among species that kind of give rise to differential abundances in a community. And those fitness differences might come about in microbe-microbe interactions, microbe-macrobe interactions, or microbe-environment interactions where you have maybe like pH or salinity selecting for organisms. And then there's also indirect interactions that you might think where maybe like a mutualism allows a species to survive in one environment and compete, whereas in the other environment, when the mutualist is not there, they are a very poor competitor. And so we think of these as being very deterministic with respect to a species identity. Next slide, please. The next of these four processes is diversification, which we see as kind of maybe a combination of stochastic and deterministic influences. So you might have kind of something that's very deterministic with respect to a trait that you're interested in. Maybe organisms are changing in response to sort of sunlight, and they're getting more pigment to deal with the influence of the sun. Or maybe you have stochastic species that arise kind of not necessarily linked to the trait or the outcome that you're interested in looking at. And you might hear me use diversification and speciation interchangeably. And again, this is just like kind of to point out that like this is linked directly to what RA3 is thinking about. These, kind of all of RA3 is like couched within this framework as well. And so it's, a great opportunity for crosstalk, I think, between us and RA3. Okay, next slide, please. Okay, so dispersal. We tend to think of dispersal as being primarily stochastic in the systems we're thinking about, but it can also be deterministic. So an example of stochastic dispersal would be a rain event not related to the species identities. It just comes through and maybe sweeps a species off its feet and into a new community. Or it could be deterministic. Perhaps a species has a flagella that allows it to move into a new space, or maybe when that rain event happens, it actually can create an ooze and be able to move to a new location. Okay, and the last pillar we're thinking about is drift, and this is totally stochastic. It's not based on species identities at all. It's just the idea that if you have a demographic difference, maybe one species is less abundant than another species, you might lose it simply by chance, and it's a higher chance to lose a less abundant species than a more abundant species. Okay, next slide, please. So how do we quantify these things? One way to quantify them, and there are many ways, is this method that's very closely linked to these four kind of processes, which was developed by James Stegan and colleagues. Next slide, please. And I just want to take a step back and think about the assumptions of this technique, because there are a lot of assumptions, and I think one opportunity we have in eMERGE is actually to test some of these assumptions. So this technique was developed with 16S sequencing, amplicon sequencing, and now we're applying it with metagenomes, which is really great because we actually can start to ask some of questions about these assumptions. So the first assumption, next slide, please, thank you, is that you assume a meta community. So a meta community is simply a collection of communities in a region that can exchange species through dispersal. You assume a meta community where dispersal is much greater than speciation. Next slide, please. So this means that new species in the local community come from dispersal rather than speciation, and that species go locally extinct primarily due to selection and drift. Obviously, this is a big assumption, and I think it's a great opportunity for us to try to test and ask questions about whether this is true. But it does lead to the fact that now we are distinguishing between 3 of the 4 rather than 4. So selection, dispersal, and drift. Next slide, please. The second big assumption is that we assume that we can see evidence of selection in the phylogenetic tree, which is the idea that niche preferences of near relatives are more similar than niche preferences of distant relatives. The idea being that the phylogenetic tree is actually telling us something about the selective history that the community has experienced. Okay, next slide, please. Now, once you have those assumptions in place, or you have convinced yourself that those assumptions are relevant for the scale and resolution at which you're monitoring the community, you can do this kind of 2-step process. The first step is you distinguish between selection and dispersal and drift. So the way you do this is by asking if communities are phylogenetically over-dispersed, under-dispersed, or neither. And so that's, next slide, please. Yeah, so if you find that your communities, your members of your community are more closely related to each other than you would expect by random chance, then you say that this is an abiotic or environmental filter that is kind of pushing all of the organisms to be more similar to each other. Next slide, please. If you see the opposite, that organisms are more distantly related than you would expect by chance, you say that this is probably because they're trying to compete with each other, and in that competition have outcompeted their near relatives. And so you say competitive exclusion has happened. Next slide, please. If you find that it's not different from randomly generated phylogenies, you say, okay, so selection is probably not the primary force going on here. Instead, we're going to start looking at dispersal or drift. Next slide, please. So second step, distinguish between dispersal and drift. So now, instead of looking at phylogenies, we just look at the kind of beta diversity of like membership and abundance. Next slide, please. And so if that's less different than you would expect by chance, and that means that the communities are more similar. So you think that is probably happening because dispersal between those communities is happening at a really fast rate. And so the communities look pretty similar to each other. On the other hand, next slide, please. If it's greater than you would expect by chance, you say dispersal rates are low. So there are not, these communities are diverging from each other because the dispersal between the communities is not keeping them similar to each other. And so instead, the communities are diverging due to drift, where you may lose species due to their abundances. And final slide. Yeah, so and then if it's not different than chance, you say drift is acting alone. So anyways, this is one way, it's not the only way, but one way to be thinking about these communities. And now I'm going to pass it over to Jessica to talk about assembly in permafrost specifically.
Thank you, Hannah. Before we do that, actually, Derek has a question that I think you would probably like to address. So he says, have you thought about specific hypotheses or data analysis to address those assumptions for meta community?
Yeah, we have actually, Dylan and I have been thinking a lot about this for the temporal paper, and thinking about how we might, for example, ask, so I guess is your question about like whether the communities are connected to each other, or is your question about like the second assumption?
Both. You said that you were trying to address some of the different assumptions that the data present for eMERGE and isogeny presents a great framework. So I was just kind of wondering if what specifics you guys had been thinking about.
Yeah, so I think we tend to think that these are part of a meta community. That is less, I think, of a question than whether or not they are, their speciation rate is less than their dispersal rate. And I think that is something that we can ask by looking at the rate of new species that we've seen over the 10 years using kind of like RA3 techniques. And also we can, ask how much these traits of interest, say methane oxidation or something like that, I'll just use that as an example, is actually tied to the phylogeny that we are using. So if we use a core phylogeny of core genes in these MAGs, do like methane oxidation genes track that phylogeny well or not? And so if they don't, then we can say that this is probably not telling us much about methane oxidation, but it might be telling us about other traits that are closely linked to phylogeny, the phylogeny we're using, because of course you can use different phylogenies.
Yeah.
I think this kind of gets back to a question that I posed to Matt yesterday, and I think his cutoff was the 95% ANI. And so how I guess this all goes into developing with these systems and developing using this technology, but how confident are you that you can really address the different speciation events versus the dispersal rates and things like that using the MAGs over the 10 years and things like that? And does it matter about like how well those are? I would assume it matters how well those are assembled over those 10 years and things like that. So I guess
definitely matters how well they're assembled. I think I'm not so worried about the quality of the assemblies. I think one question is whether we, like you draw a line in the sand and kind of a cutoff for what you define as a new species, right? And so if we use that 95% that Matt brought up yesterday, then what we need to be able to apply this technique is to know whether at that 95% cutoff, whether new species have arisen over the last 10 years. And if they have, then I think, one way you could do it is to try to first identify where the new species are on the tree, and then ask the question of whether or not those are the reason, those tips are the reason that you're seeing, so-called high dispersal, right? Because in this technique, what would happen if you had new species coming is you would mistake them for kind of dispersal happening, when in fact they're actually new species originating over the time involved. So I think the way to handle this is to turn it into a 3-step process, where the first step is make sure that you understand the role of diversification. Once you have ruled that out or kind of accounted for that, then you can go ahead and look at selection, dispersal, and drift. That's what I'm thinking right now. But if anyone has other ideas about this, I'm all ears, because I think that it's something really new and interesting that we could be doing.
And I guess, thank you. I mean, this is a great discussion. I don't want to keep taking time away, but how important will it be to say have transcriptomics or proteomics for those mags? Because I think it would be difficult in my mind, just like a loss of function mutant or something that's not expressing, say a given function. So will you really need, say, the transcriptomics or proteomics to have a better inference on whether or not this is just a random mutation, like a loss of function mutant? If it's just not expressing something, I guess, how does that really actually come into the definition of speciation or things like that?
Yeah, I think that's a great question. I think so. If we have a transcriptome or proteome, we can actually start to say whether or not the selection is not acting on the genome, right? It's acting on the phenotype, and so the transcriptomics, proteomics, that comes into bringing us much closer to phenotype as opposed to this genome. And so I think it's kind of the struggle that we all deal with when we're working with this data, which is what is actually being seen versus the kind of the proxy for the proxy for the proxy, right? To bring back Sarah's point.
The great news is that it's so much better to be using the type of data that eMERGE has collected than the typical type of data that, or it's also harder, it turns out, but than the typical type of 16S data, where you're actually having to make some pretty large assumptions and doing some kind of asking questions about clustering versus in like environmental features. But this we can actually test those things more explicitly, whether the clustering means anything to the kind of the niche preferences. Okay, so I will keep us moving along. Hannah, I was just pointing out in the chat to everybody that when you guys just said assembly a few times, you were actually talking about assembly of the mags and not assembly of the communities, and that might have been confusing.
Yeah, hopefully I interpreted Derek's use of assembly correctly, not sorry if
yes.
I did.
I also assumed he was meaning that. And so I'm going to ask this question, or we're all going to ask this question together. Is permafrost a special case for assembly? And so Hannah just gave you that really nice primer on kind of the background of assembly and how it's come about. And so I want to talk to you about microbial communities in permafrost and how thaw might affect them. This is a work that I'm doing with a pretty large group of people as we've developed this kind of thing called the Permafrost Microbiome Network. So if people are interested in getting involved with us on that, I hope that after we get this paper resubmitted soon, that we'll actually move on to other things. And so it'd be really cool to have more thoughts there. But I would like to point out that we do have some other eMERGE, not just members, directors here on this. So Virginia's on this, for example. Okay, so I wanted to take a moment and have like a little chat storm. So I'm going to ask you guys 2 questions, and I want you to write your answer in the chat, but then don't hit enter until I say so that people have a moment to think. Okay, so from what you heard about from Hannah and what you heard about from Kelly and Malak the other day, and then Owen yesterday, what are some abiotic-biotic interactions that shape microbial community composition? So go ahead and write those, but don't hit enter yet. Okay, go ahead and hit enter. Yeah, seeing a lot of pH coming up, a lot of temperature. Those are things that we notice are really changing in the mire, right? Ooh, pressure and salt. Yeah, I think that's actually particularly relevant in permafrost systems. Changes in plant species, changes in moisture. Yeah, so these are great. These are kind of like our classics, right? So it's perfect. Redox conditions is on here too. And then let's talk about biotic ones. So what biotic interactions are going to shape microbial community composition? Jeff, don't wait. Oh, Vivi, by the way, brought up soil organic matter composition. Good job. Okay, yeah, let's hit enter now. Great, so I'm seeing predation, facilitation, trophic structures, syntrophy, litter inputs, plant and predation, viral predation. Yeah, so we've all been listening the past few days. That's great. My microbial growth efficiencies. Yeah, absolutely. So like who can actually get the resources? That's like classic community ecology right there. Great. So I just kind of wanted to have a moment where we like kind of shake it up and connect that with other parts of what we've been learning about. And then I think that Hannah and I are transitioning quickly enough between us that you probably will not have also forgotten that there are a lot of stochastic processes underlying assembly, but we should also probably consider those. And, people don't often actually do that, especially in microbial communities. It's increasingly becoming more common for people to be putting an assembly framework on microbial communities, but it's still very uncommon in permafrost systems, although it is done. Before I so I wanted you to like ruminate on what factors might underlie some of that stochasticity. And I wanted to tell you a bit more about permafrost microbes too, while I had the stage here for a moment, since that's probably not something that everybody thinks about all the time, or like the special case of those microbes entrained in that frozen soil. And so I do specialize in that actual frozen soil. I know, great photo, right, Katie? Sorry, Katie, I noticed now that I've adjusted it, it's a little bit large, it's like stretched, but this is an awesome photo of Katie in 2018, who's on the call, when she was doing some work as an undergrad up at Stordalen, and she was doing the gas traps on this new thaw pond on Stordalen. And don't worry, Katie, you're going to get to see a lot of this photo. I apologize. I've like used this as a background for other talks, and I'm like, well, I hope Katie doesn't mind. Well, I asked you about it, but now you're going to see yourself a ton. Okay, so what do we know about permafrost microbes? And maybe just quickly, I should mention that I did my PhD thinking like entirely about the specific like permafrost frozen system and how it might be feeding back to climate. And I started that work like before I got the chance to see all these awesome papers that came out of eMERGE. And when the eMERGE papers came out, I was like absolute fangirl, because I did all of my work like characterizing carbon chemistry and characterizing microbial communities in permafrost, and then thawing them and measuring functional potential and measuring gases and things. But this like idea of a thaw gradient is just so genius, right? So you guys are, everyone here is on this awesome project that I like totally fangirled about, and now I get to like fangirl and be here, so I'm pretty stoked. So anyway, okay, what do we know about permafrost microbes? We tend to think they're selected for survival under really harsh conditions. And so here is a study that worked on this. So this is the permafrost tunnel in the cold regions permafrost tunnel in Fairbanks, or outside of Fairbanks, Alaska. And in this study, Rachel McElprane collected metagenomes for permafrost of 3 different ages. So a 19,000-year-old permafrost up to a 33,000-year-old permafrost. So pretty old. And for those of you who can look at heat maps quickly, that's on the right. But if you can't, I'll just tell you what it says. So they kind of came to the idea that permafrost microbes are employing these long-term survival strategies and have traits that may enable survival. And examples of these traits include scavenging detrital biomass, horizontal gene transfer, chemotaxis, dormancy, environmental sensing, and stress response. So that's just one example. But then we also know that active layer microbes and permafrost microbes aren't the same, or we know that in other systems. Actually, I don't know if we know that here yet, Stordalen. We actually do know it across the street. I'll show you that in a moment. Okay, so here's from my site from Alaska, up at Sagwan Hills, which is north of the Toolik Lake Biological Field Station. Thank you, Martinez, perfect. So way up north. And here what I was looking for was if you see, this is old sequencing, this is like 454 pyrosequencing, but if you see a line going all the way through here, that's an OTU that's shared between all the layers. So we have organic active layer, mineral active layer, and then this is a mineral permafrost system. But you'll see that a lot of them aren't shared in between. Some are just shared between, say, just the two active layer soils, and some are just shared between, say, like all the mineral soils. And if it's easier to see that in a Venn diagram, then you can see here that the active layer here and the permafrost have pretty distinct communities. So those are shared OTUs. We also know that diversity declines with depth in these systems for bacteria and fungi, and increases for archaea. So looking at the same, I did the cardinal sin of a line through two depths, but you'll have to forgive me for that. And I apologize, neither of these two figures are published. I have like one PhD chapter that I don't know if I'll have to give up on at some point, but maybe someday it'll still come out. You see a decline in diversity with of bacteria and fungi, and an increase in diversity with archaea. And so these are, some pretty deep permafrost soils. We have a much shallower active layer up there. And we also see changes in functional diversity. So I think this is where we start thinking about, well, how are these things going to function after thaw? So here I'm showing you some proportion of substrates used. I did this, what they call a biolog assay, which some of you might have done. And I ran this out over 3 months and actually was able to track growth rates and obtain kinetic parameters with some modeling. But here, I'm just showing you the percentage of substrates used. And you see at 1 degree versus 10 degrees versus 20 degrees, a really different substrate use profiles. So the permafrost is not using very many different types of substrates at 1 degree. They ramp up at 10 and 20, which was pretty surprising to me. The organic active layer actually keeps the same growth rate throughout keeps all the same number of substrates used throughout those temperatures. Growth rates did track temperature, but the active layer was actually more competitive as well. So another thing we know, and you all know this very well, is that when permafrost thaws, the composition of the microbiome does shift. So here's I think the first example I know of this, which is from Rachel McElprang as well. And she has here in this circle active layer soils and permafrost soils. And here's the frozen starting points. And then you go to day 2 and day 7. So it's a pretty short incubation, but she saw large shifts in the microbiome that don't seem to be converging yet, but of course it's only 7 days. Here's a longer term study. So this is from Sylvain Monteau across the road from us. He's at Storflocketmeyer, and this is from a snow fence experiment. And so first I'm just showing you the controls. So active layer, what they're calling the intermediate layer in their soils. So the bottom of the active layer, and then the permafrost soil. And then after, I guess it's 10 years of thaw at the point where they studied this, they're seeing large shifts in the permafrost community. The intermediate layer actually starts to converge with their active layer soils. And so that's, I think, really interesting finding, because that's actually in the field rather than in the lab. And then of course, you all know this very well, because we know that the microbes under the different the 3 different thaw stages have really different compositions. And also we know that microbes in permafrost encode for a really diverse suite of functions, and that they are different in the different sites. And we've heard a lot about that this week. So I'll keep telling you about other permafrost sites mostly. But of course, we want to be able to predict how, we don't just want to always be observing that community composition shifts. And I think that goes along with us wanting to be able to predict greenhouse gases, but it's also just the evolution of the field as well, that we're moving beyond just being able to sequence and to be able to actually understand. So So one thing we know, and you all just told me this, right, is that microbiome composition is driven by abiotic conditions. So here's an example where I took those mineral permafrost soils I told you about before and incubated them for 90 days under oxic and anoxic conditions at 2 temperatures. And not surprisingly, I see really different compositions. We also know that there's biotic conditions and interactions that affect the communities. And Moira went over this, I think, on day 1. But where we see that the microbial communities are different depending on which plant compartment you're looking at and also which habitat you're looking at. And so then, what else? So I think then this kind of gets us transitioning to, okay, well, what don't we know? We know from what Hannah just told us that it's probably very important that we also care about stochastic factors, but we don't actually know a lot about them in permafrost systems. We know some things though. So one, first of all, does it matter whether a microbiome is shaped by deterministic or stochastic processes? And so I'm just showing you like kind of a little cartoony example of why it might. So if this is selection by some environmental filter here, we have a trait of interest that of course is making it through because this is selection, it's being selected for in the community. Other members are being lost because of the selection. But if we think of stochastic processes, so here I'm showing probably drift because we're having a random loss of those blue members. You could also have instances where dispersal actually introduces members. So you could have a gain in function. You can have also where instances where dispersal kind of overrides other things, other factors, and ends up with diluting your functional capacity of that community. So then what evidence do we have for stochasticity following permafrost thaw? Well, to my knowledge, we only have one piece of evidence so far. So this group will be doing pretty cutting edge research. And this actually also comes from across the way at Storflocket. This is Stacy Doherty, who many of you know. She did a master's in my lab and finished up in 2020. And she looked at Storflocket actually all in the control plots because silly me, I did not send her with the extender for the core because I didn't even realize like how deep permafrost was up there and I was on maternity leave. I'm like, oh, don't take it, it's so heavy. So the control plots were sufficiently thawed though, so she didn't need the treatment plots, but it was pretty silly of me. Now I know better. So looking at the active layer down to 50 centimeters, this is what we're calling the transition zone, which was new active layer within the last 10 years based on their probing data. And then some still frozen permafrost. And she sees in frozen permafrost, so she used that technique that, that Hannah was talking about, the beta null modeling technique, and she sees drift and dispersal limitations, so these stochastic factors, also selection though in the permafrost. So these are kind of what we expect. We expect selection for those conditions, a dispersal limitation, this all makes sense. Then immediately after thaw, she's seeing a really big increase in drift and then also seeing dispersal limitations still playing a part. So I find this fascinating and I really want to know if this is something that we can recreate across the world or not, or if this is just a particular thing at Storflocket or a particular artifact of this method of modeling. It's really fascinating. And then over time, you start getting more selection playing a part. And then in the active layer, we're seeing all selection. Okay, let's see. So, and then I guess another piece of evidence, although I'm running short on time. So I'm gonna kind of haul through these, but there is more evidence for missing functions after thaw. So this is a study done by Christian Knobloch, 7 years of a permafrost incubation, where some of their cores never produced methane until they inoculated them, and they couldn't detect any, they were just doing like qPCR with MCRA genes, but they didn't detect any methanogens. And then they added some soils that had been methane active, and then they saw methane. And so I think that's a great example. Sylvain Monteau also did some types of those experiments with permafrost out of the permafrost tunnel and also sees like really large increases in CO2 and things once you add in microbes. Okay, so thinking about where we started, thinking about the folks working on this paper, now we thought, well, okay, we want to be able to make a contribution as permafrost microbiologists to thinking about what this post-thaw microbiome is going to do and what the climate feedbacks are going to be. But we don't currently have a framework for predicting what the composition will even be. So it's hard to predict what it will functionally do. And so we thought we could try to apply this assembly framework. At this point, it's all conceptual. And we, through a series of literature reviews and really deep reading on assembly, have put this together. And so we are positing that the pre-thaw microbiome is important to consider. Considering space and time is going to be important as you consider the community, and then also disturbance intensity. So just want to show you this. So this is our thawing permafrost landscape over time, right? And so after time, we're getting this post-thaw community. Now I'm going to dive into some of the mechanisms we think are at play in the post-thaw community. So this is like my absolute favorite figure, and this is also by Victor Leshik, but it is complicated, so I'll try to walk you through it. This also is functioning over time. We just kind of, I took the figures apart, so you're kind of losing that now, but this is happening over time. This is the post-thaw realized community. This is an intact community. So you're seeing movement of microbes, like an introduction into that post-thaw community with the arrow movements. You're seeing drift with the X's, and then you also see like survival. So for example, surviving through environmental filters. You're also seeing diversification here, and then a lack of survival. So now I'll walk you through the thaw stages, since I've talked about what those arrows and stuff mean. So here's some of our hypotheses that are coming out, and it's all very, kind of convoluted how we got to these, but now I'll just throw them here for you now. So here in intact permafrost, we're saying we have good evidence that the community is shaped for selection for survival and growth under these frozen conditions, and then also dispersal limitations. You don't see the dispersal. In these early thaw stages, assembly of the post-thaw microbiome is dominated by dispersal and dispersal of new members and drift. So drift becoming very important in that post-thaw, like immediately. And I think we've also come to believe that the disturbance intensity, so here I'm showing maybe just an active layer deepening versus a thermokarsting versus a retrogressive thaw slump where the whole landscape is just kind of falling apart, are going to affect the amount of drift and dispersal you would see. And then in this final thaw stage, like as thaw advances, I don't know the length of time that is necessarily, but dispersal and drift will of course continue. They're always part of community assembly, but selection will start to become more important, and also diversification will start to become more important. And so you see the microbes need to make it through their environmental, biotic, and abiotic filters. But of course, those prior interactions between disturbance intensity and stochasticity are going to affect that starting point of diversification and selection. And so that might actually lead to something Hannah mentioned, I think, in the chat earlier in the week, like alternate stable states is a question that a big question I have about post-thaw assembly of microbiomes. Okay, so here's the main takeaways. We want to use an assembly framework to consider the impact of initial composition, disturbance intensity, and space and time. And we hope that this will allow us to make some predictions about composition and function of the microbiome after thaw. In the short term, we predict that communities, they'll be unpredictable. Great. Okay, well, we think they're going to be unpredictable, both in composition and function. And so we hypothesize that particularly in high disturbance, assembly is driven by stochastic factors, and that there will be implications for modeling greenhouse gases and plant feedbacks in short term. And then under longer term, we predict, or probably even hope, that communities are selected for the new conditions, and that will lead us to easier ways to predict who's there. And then dispersal will again continue to play a large role, and there may be alternate stable states if drift dominated early. So, okay, I think I didn't totally give us exactly the right amount of time, but we're not too far off. So we're going to do a group exercise. I guess I didn't ask you, Jin, if we have breakouts already created.
How many do you need?
4, is that right, Hannah? We're having
Yeah, we
people
could
just
do
go
4
in.
or 8. 8 would make groups of 4, I think, ish.
Oh yeah, let's do 8 maybe.
Okay, and do you want to assign manually, let participants choose room, or assign automatically?
Let participants choose the room. So groups 1 and 2 will be selection. 3 and 4 will be diversification. Did I get that right? 1, 2, 3, 4. Yeah, 5 and 6 are dispersal, and 7 and 8 are drift. Sorry, should have written something on the slide here. So choose a breakout room for one of the 4 processes and develop a hypothesis of how it might be at play in the microbial communities in the mire. And if you can just put your notes and thoughts onto that Jamboard that we have, that'd be great. And then we'll give you 10 minutes in your room, and we'll just kind of come back for a bit of rapid fire with our last 4. Hannah, are you typing this in the chat?
Yes, I am. Sorry, you can probably see me
Fantastic.
typing. Yeah.
No, it's just great. Okay, perfect. Should I stop sharing, everybody? Maybe I'll leave this for people to see. Okay, so your task is to develop a hypothesis of how the assembly factor you pick might be at play in the microbial communities in the mire. All right. All right, so I appreciate that everyone got some awesome notes up. So I'd love to hear from maybe just like 30 seconds we could probably pull off from each group. So what did the selection group have to say?
Sure, I can read our soundbite. Our end soundbite, we said, cumulative effects of climate, thaw, hydrological, vegetation, geochemical feedbacks increase the role of selection in community assembly over decadal scale thaw gradients, like Stordalen.
Cool. I like how you made a brand new very hyphenated word. Great. Let's see. How about the drift group? Oh, actually, you know what? Sorry, I'm going to go in more of an order, like, okay, going along not just the next one I see. So how about like the diversification group? So we're getting a little bit more stochastic.
Did we have a diversification group? I don't see.
Yeah, they're on the next page of that because oh wait, no, maybe 2 groups did drift. Ooh.
We have 2 drift groups.
Interesting. Okay. Wow. All right. Well, you're like, who cares? Doesn't fit in Hannah's model anyway. How about the dispersal group?
So I'll talk. We were talking, I brought it up about plant roots and the role of plant roots rapidly exploring both a seasonal thaw zone and also a long-term extended thaw zone, and how that affects either physically or through the environment microbial dispersal and how fast microbes can move. And then there was, Sarah talked about flagella expression and sticky surfaces controls. And
cool. Thanks, Steve. You must, I need to catch up with you because I have like a whole project on that, on plant roots in permafrost, moving into permafrost layers, and what that will happen, like how that will change microbial communities and carbon cycling. So you guys are like right on to my thoughts there. Then we have 2 drift groups. So let's start with group 7 and then we'll go to group 8.
I can talk about it. So in group 7, we talked about how we think drift will be more at play earlier on. Oh, sorry, when Virginia made that comment, it made it really tiny on my screen. We think that drift will be more at play earlier on in the thaw process, and then also because drift is a stochastic process, that it will vary depending on the nature of thaw. So when we see thawing, more of like we saw in that picture that Jessica shared from the pond, where it's more erosional and kind of maybe happening quickly, that we'll see more drift at play, where if it's a slower thaw transition and there's not a lot of like material mixing and things like that, that drift and like that sort of randomness may not have as be as present.
I would like your group to be a reviewer for our paper because that's what we are trying to put forth, and we'll see if it goes. All right, and then group 8.
I think I'm drawing the short end of the stick here. So I will try to report out and hope there's not a screaming child that mutes me. But I don't know that we were able to get to an actual solid hypothesis. We just kind of discussed some of the different aspects of genetic drift that we thought could or could not be important. And so just the loss of gain of functionality of the genes, say something like changing, like the other group brought up with changing redox conditions, changing thaw, and how you could then change the ability to consume a substrate or not, and how that could have a direct fitness impact. Something losing the ability or potentially gaining the function of like quorum sensing or extracellular other signaling type molecules. And then we thought that the population size would probably matter as well. So thinking about isolating key communities or populations and like pore space or something like that, and how that could then impact the ability to counteract stress levels and substrates and things like that.
Thanks, Derek, for taking it for the team there. I actually wonder, and it sounds like your group had maybe a lot of people familiar with population genetics and the thought of drift there. And so I almost want to put like the first half of your thoughts into the diversification bucket.
Sure.
Because, yeah, and then the second half, I also agree though, the population size mattering is, big on a big thought with drift that I did not get into in the talk. But Virginia and Hannah and I were just discussing that perhaps a deep dive is necessary to get into more of this. We know it was like such a whirlwind. So we will work on that. I cannot make any timeline promises at this very moment, but great. Well, then